pleasure to introduce is our departmental seminar speaker, Dr. Richard Young. He's a professor at the Whitehead Institute and MIT. He's at MIT for the entire time he's been a professor, but before that, he did his undergrad studies at Mediator University. He's a grad student here at Yale in Joan Stites' lab. And he also conducted postdoctoral work in Switzerland and at Stanford University. Dr. Young is really a scientific pioneer and leader, and his research over the years has spanned from viruses, bacteria, and yeast, but most recently, his lab has focused on gene regulation and transcriptional control in mammalian systems. In the last decade, his lab has really of super enhancers in the control of cell identity, and his lab is really a pioneer in thinking about how the phase-separated condensates can really control transcription. And that's a topic I'm sure we'll hear more about today. I think what's really remarkable about Dr. Young's work is that it's very far-reaching, touching on not only fundamental biological principles, but also mechanisms of human disease, with lots of contributions to medicine and pharmaceuticals as well. His contributions to science have been recognized by many. He's a member of both the National Academy of Medicine as well as the National Academy of Sciences. He served as an advisor to the World Health Organization and to the NIH, as well as to many other scientific societies and journals. In addition, he's founded and advised many companies in the biotech and pharmaceutical industry. And before I hand over the floor to Dr. Young, I do want to mention that he currently co-teaches an MIT class that's specifically about the biology of COVID-19 and SARS-CoV-2. And this class features guest lectures from leading virologists and immunologists. And anyone can access the recorded lectures online. And Dr. Young was telling me about how many people did you say? I think 5,000 plus people are live streaming it. And more people are probably listening to the recorded lectures as well, which is fantastic. And so far the lectures that I've listened to have been really informative and accessible. So I'd encourage you to check out those lectures if you have the time as well. And with that, I'll let Dr. Young share his thoughts with us about biomolecular condensates in gene regulation, cancer, and medicine.
Mina, thank you so much for inviting me to come virtually to Yale and talk to you about our stuff, and to give me an excuse for the first time to put on a jacket and dress up for a public event. So I'm going to share my screen now and just check to make sure that we have the right slides up. Yep, I think we do. So as Mina said, my topic today is going to be on nuclear condensates and gene regulation, cancer, and medicine. And I'd just like to start by giving a shout out to my mentor, Joan Stites. Joan taught me many things as a grad student. She taught me about the quality of science and what that meant. She taught me about collaborators and collaborating and the importance of engaging people in your work. She also taught me to be brave. At the time I entered her lab, she was shifting from her focus from the mechanisms by which ribosomes initiate and the mechanisms by which bacteria transcribe ribosomal RNA operons to this field of ribonucleoproteins, ribonucleic particles that emerged as an important beginning to our understanding of how spliceosomes function. And Joan, thank you. You've been a wonderful friend. So the topics I want to cover today are three. I want to review for you how we think of what is termed biomolecular condensates, and where we are in that space. I want to talk to you about dysregulated transcription in cancer and how that's connected to condensates. And then I'm going to tell you about a discovery we made recently about cancer drugs, successful cancer drugs, and how we think they work. I think they're working in a manner that was not anticipated and changes my view about how all future drug discovery will take place. One of the things that's been so inspiring about structural biology is you can actually see how proteins and their substrates interact and how proteins change conformation to exert work. This is a picture of the RNA polymerase II multi-subunit molecule out of Patrick Kramer's lab. It's absolutely gorgeous. It's used to consider hypotheses about how transcriptional control works in mammalian cells. The issue, though, having been inspired in our understanding about how much of molecular biology works by these structures, the issue is that a good fraction of the proteome does not conveniently form such structures. Indeed, probably 25 to 30% of the protein coding capacity of the genome encodes what we think of as structurally unstable, intrinsically disordered regions, or IDRs. So for example, with transcription factors, and our genome encodes about 1,000 transcription factors, many of those that have been successfully crystallized give you the atomic structure of the DNA binding domain. And just gorgeous resolution. You see this work from Nair and Burley from way back in the early 2000s of the Micmacs heterodimer. In contrast, however, no activation domain of any transcription factor encoded in mammalian genomes has ever been successfully crystallized. And yet all transcription factors have them. All transcription factors have a DNA binding domain and they have an activation domain. So what we know about the activation domains is that you can generate from NMR ensemble structures, and it appears that these are very dynamic in their interaction with what we term as co-activators, the molecules that are necessary bridges between transcription factors and the rest of the transcription apparatus. What's emerged in the last few decades and has really been encapsulated in an important way in biology by Cliff Brangwine, Tony Hyman, Mike Rosen, and others, is that the IDRs have a property inherent to the physicochemistry of polymers, and that is they have the capacity to phase separate and to form what we term biomolecular condensates. Let's talk about these for just a moment. Polymers can overcome the entropy of a system by producing a large number of weak interactions. And what you see in the top right panel is a theoretical diagram of a phase separation event that's occurring where the two parameters we're measuring are concentration and temperature. And what you see at the extreme right is what we call the demixing of, say, the green droplets relative to all the other components of the system. And as I mentioned, proteins often have both structured and unstructured domains, disordered domains. Sometimes they're oligomeric, small domains of structure linked by additional domains of the same sort. And the features that you get then are that the structured domain is capable, as we well know, of high affinity interactions with a defined substrate. In contrast, what we've learned is that these disordered or oligomeric domains can have very different stoichiometries in terms of their interactions with other proteins. They're interacting through low affinity interactions at high valency. And I'm noting in the bottom right, really the leaders in this arena that have inspired me to think more deeply about how this might apply to events in the nucleus. What's emerged really in the last 2 to 3 years is the understanding that every cellular process that's been subjected to serious genetic and biochemical analysis over the last half a century occurs to some degree in non-membrane compartments that are now called biomolecular condensates. That term, biomolecular condensates, is not meant to imply that the mechanism by which there was a formation of a compartment is through phase separation, but as we understand it right now, what is called liquid-liquid phase separation, where two liquids can separate into a dense body and a surrounding body that has less density of the specific RNA, protein, or DNA of interest, that this likely happens in cells. Because these are first order transitions, you can really only demonstrate that they happen in very purified systems in vitro. But the features that are observed in vivo are features that one would expect for liquid-liquid phase separation. And so at the moment, most of us think that's the underlying physical chemistry that produces these compartments, which can have long half-lives or short half-lives, depending on the dynamics that are occurring in the compartment. So many people ask me, so what? What do you really learn other than there's some compartmentalization that isn't really solved by these powerful tools and thinking that we've had in molecular biology? Well, it makes a difference if you're going to diffuse around and find your partners if you have several hundred partners engaged in a particular function. And it is of interest that there's a property in condensates called partitioning, where proteins, DNA and RNA, have preferences for the bodies in which they like to reside. You can move beyond thermodynamic modeling of equilibrium conditions to non-equilibrium models that better reflect the functional dynamics of cells. And there are disease mechanisms that are really not well explained by conventional lock and key models. And so there are new theories that are inspired, new theories that are leading us, for example, to imagine new ways of developing therapeutics for various Mendelian and cancer diseases. So there were two mysteries in gene regulation that particularly interested me a few years ago, in 2016, when I began thinking more about the possibility that condensation was involved in gene control. One, what I already alluded to, that the human genome has thousands of transcription factors. They consist of these two domains. The activation domain's interchangeable. You don't have to have a specific activation domain on a transcription factor. And this deep knowledge we've had of the DNA binding domains actually led to transcription factors being classified by their DNA binding domain, because the activation domains, really the specific sequences, don't appear to be terribly relevant to the function. The other thing that interests me is, as Mina pointed out, we observe these clustered enhancers. We and others were surprised that these clustered enhancers are associated with genes that we regard as playing the most prominent roles in normal cell identity. And in dysregulated cells, like in cancer cells, we routinely find exceptionally large clustered enhancers that are not in the cell of origin, but do occur at the driver oncogenes. These can interact. Super enhancers that occur across an individual chromosome create a propensity for those portions of chromosome to interact in cells. And finally, Wendy Bickmore and others have observed that, in contrast to the model we've always had about how enhancers loop to promoters and thereby activate their target genes, the enhancers actually turn out not to need to get any closer than about 200 nanometers to do that activation. And that's quite mysterious. So these mysteries really led us to think about the possibility that phase separation is involved in transcriptional control. And I'm just showing you some of that evidence that is already published. In the top left, what you're looking at are puncta that are generated by the transcription factor OCT4 and the co-activator subunit MED1. These have been tagged with monomeric EGFP in their normal endogenous locus, and they're being expressed at normal levels. And what they do is they form these large puncta, about 300 nanometers in size, at the superenhancers that occur in those cells. Here we're looking at murine embryonic stem cells. And what you can see in the little movie in the center is these are very dynamic. They appear and disappear. And this formation and dissolution occurs, depending on the site, in second to minute time frames. What I'm showing you in the bottom is also some published data that simply says that if you now purify these transcription factors, and we've selected a variety of transcription factors that have been very well studied either in developmental biology or in cancer, and have asked, do the activation domains of these transcription factors confer on the factor the ability to separate droplets in vitro? And the answer is they do. And those droplets are liquid-like. They have all the behaviors of a liquid. They will fuse with other droplets. They exhibit less than 1 minute timescales when you photobleach them and allow factors outside the droplet to exchange. So these look like they are phase-separated liquid condensates. And what's remarkable and exciting to me is that any mutation in these activation domains that's previously been shown to have adverse effects on transcriptional activation has adverse effects on condensate formation. And moreover, the ligands that nuclear hormone receptors, for example, ER bind, ER binding to its ligands is necessary for optimal transcriptional activation, and similarly, ER liganded with estrogen is essential for the formation of these condensates. So there's this beautiful correlation that suggests to us that in vitro, there's liquid-liquid phase separation associated with these transcription factors interacting with their cofactors. And in vivo, we can observe puncta whose behaviors are also consistent with liquid-like droplets. So the model that's emerged from this and much other data is that there are phase-separated condensates that occur at superenhancers. What is happening is the DNA binding factors are being crowded on the DNA. That's contributing to phase separation because it's driving those factors across the threshold for the concentration at which phase separation would occur. And the intrinsically disordered portions of those proteins are engaging in very dynamic interactions with mediator and other co-activators. And we can actually see with Ibrahim Sisi clusters of RNA polymerase molecules, as many as 80 RNA polymerase molecules at a time coming in a condensate to fuse with these sites. That's in stark contrast to any textbook models or any models I've ever driven in my papers where we have single molecular components coming in sequence to interact with these other molecules. What's remarkable about this model is it helps explain all the mysteries that have really confounded my field for many decades. How do these transcription factor activation domains function? How is it possible that hundreds or even 1,000 different transcription factors can interact with the same set of subunits in the co-activator complex we call mediator? And why it is that you can measure distances between enhancers and promoters of 200 nanometers or more and obtain activation. More recently, we've become interested in heterochromatin, and some pioneering work by others has argued that the heterochromatin factor HP1 alpha is forming a condensate at heterochromatin that gives heterochromatin a much more dynamic property than anticipated by some of the literature. Typically, heterochromatin is referred to as closed chromatin, and euchromatin is open chromatin. But in this new view of condensates, it's not the openness that has to do with regulation, but in contrast, partitioning behavior. So I'm just showing you here a small amount of data from work we just published last month, actually come out officially in Nature, I believe tomorrow. And what we found is just following on the standard molecular biology model, where heterochromatin is a nucleosome containing material that is modified by the Suvar39H class of proteins to give you H3K9 methylation. That is read by the HP1 alpha reader of that modification, which can recruit DNA methyltransferases and cause 5-methyl-C on that DNA. And it's 5-methyl-C that is bound by MACP2. It's a reader and preferentially reads DNA that's 5-methylated. And so what you can see then, consistent with that molecular model, is that there are heterochromatin puncta, these are murine ES cells in this case, that contain both MACP2, again labeled here with monomeric EGFP expressed from the endogenous locus, and HP1 alpha, in that case labeled with mCherry and expressed from its own locus. And the merge gives you coincidence throughout these bodies and throughout all the cells we've examined. But importantly, we've known for about 2 decades that mutations in MECP2 produce Rett syndrome. MECP2 is an X-linked gene, and Rett syndrome it's the second major contributor to female intellectual disability. It's suffered by young girls who, at the age of maybe 4 or 5, begin to revert in their development to be less and less cognitive. And those mutations occur, as you can see in the bottom left, both in the methyl binding domain, the MBD, and in the C-terminal intrinsically disordered region. What's interesting, or what really caught my attention, is that the mutations in the methyl binding domain are all point mutations, whereas the mutations in the C-terminal disordered region are, with one exception, all truncation mutations. Both types of mutations affect the ability to form normal heterochromatic condensates in the neurons of these young girls who are so affected. And much like the model I gave you earlier, what we've realized is happening is that the truncation mutations are affecting the valence of that protein. That is the number of weak interactions that can occur. In contrast, mutations in the methyl binding domain are affecting the ability of the protein to be crowded on DNA through its DNA binding. Either type of mutation leads to condensate dysregulation. And now we think that all cases of Rett syndrome are in fact mutations that are causing dysregulated condensates. And what that does, interestingly enough, is it looks like it allows RNA polymerase II to partition inappropriately into heterochromatin and begin expressing genes, for example, genes that are close to satellite repeats, alpha satellite in heterochromatin, and that is toxic to these neurons. So let me just summarize this section by saying that every one of the nuclear activities that I've been interested in appears to occur, at least transiently, in what we would call a biomolecular condensate. These are puncta. They've been observed way back since the time of Joe Gall studying these kinds of puncta in cells. And what we believe they now are compartments that functionally compartmentalize the various functions of the cell. And they give these cells regulatory properties in addition to those that are explained by conventional lock and key models. So I'm going to move then to what this might mean for cancer. And I'm showing you here a diagram from a paper that Jay Bradner and I wrote a number of years ago, just summarizing the number of kinds of dysregulated events that occur in the nucleus. Because these are recurrent events in various cancer cells, we infer that they are important for the cancer state. And as you can see on my list, there are recurrent events that create enhancer mutations, create mutations in what we think of as the master transcription factors. Often these are early lineage transcription factors that should be shut off in the ontogeny of these cells, and the cancer cells manage to turn them back on and incorporate a more embryonic state. There's, of course, classic oncogenic signaling, dysregulated signaling pathways, epigenetic alterations, and in all tumor cells, very poorly understood, there's the inappropriate expression of a broad of non-coding RNAs. But to our view, again, one of the things that brought us into this space of considering how polymer behaviors in biophysics might contribute to the kinds of functions we were looking at, what became clear to me is that the kinds of superenhancers that T cells acquire at their oncogenes are very different in a quantitative sense than those we see in normal cells. In normal cells, we measure clusters of enhancers that are maybe 10 or 20 kb in length. But along this linear span of genome, it is not unusual to find superenhancer clusters that span 200 or 300 kb, easily an order of magnitude larger than those we see in normal cells. Another thing that we see that is really striking and suggests some kind of cooperative behavior beyond that I've ever understood is that you find recurrently in cancers small indels, really small indels, that form the binding sites for a single transcription factor molecule. And we saw this first with Mark Mansour and Tom Look in TLL, where we observed the binding site for a single transcription factor MIB causing the opening up of chromatin to form a 20 kb superenhancer that would then drive the tau1 transcription factor gene, which is contributing to this TLL cancer. A single binding site. We were able to recapitulate this to introduce that single binding site into T cells and show that that could cause the recruitment of this large superenhancer with all of its components I've shown here. The other thing that was confusing to us is why it is that the developmental signaling factors, the downstream effectors of Wnt, of LIF, of TGF-beta signaling, why it was that they all preferred to go in a cell type specific fashion to superenhancers associated with the key genes in those cells. And what we've come to understand is each of those effector factors, whether it's beta-catenin or a SMAD or another factor, have these intrinsically disordered regions like I'm showing here for beta-catenin. And what that allows them to do, what allows cells to evolve to do, is to use the same signaling apparatus, but to give you different cell type specific signaling, because these proteins partition into the super enhancers that are by definition cell type specific. So each of these three kinds of views of cancer cells is better explained, in my view, by these condensate models and regulation of transcription than more conventional models that I've used to think about transcriptional control. But one thing has remained a mystery to me, and that is in work that we have done with Jay Bradner and Nathaniel Gray for the last 6 or 7 years, where we've employed various kinds of transcriptional inhibitors on cancer cells, transcriptional inhibitors that should affect the transcriptional output of every gene, because their targets were cofactors. BRD4, I'm showing here, CDK7. These are cofactors for every gene in every normal cell. But what we were observing was that there was a therapeutic index. There was, instead of every gene being similarly affected, there was an unexpected result that was really hard to explain. And that was that you could find the driver oncogenes in various tumor cell types much more profoundly affected than the other genes in these cells. So why was it that these super enhancers would be exceptionally sensitive? I would have thought that instead they would be harder to inhibit, given the vast excess of the target proteins for these inhibitors of general transcription. And it's really to that issue that I'm going to focus the attention to the rest of my talk. So the key criteria that you think about if you are developing a drug are pharmacodynamics and pharmacokinetics. Pharmacodynamics, that's all about the density of your, in this case, small molecule. I'm showing you how hormones and anti-hormone activities impinge on the estrogen receptor. Pharmacokinetics is more about absorption, distribution, metabolism, and excretion. But in this, the concern is about tissue distribution. It is not about distribution within the cell. And we've typically assumed, because it's not easy to study it otherwise, that once a drug gets into cells, it's typically going to access most of the compartments within those cells. We wanted to know, though, if that was really true. But the first thing we had to do in cancer was just simply show that, that these condensates that we've been studying actually occur in the tissues of normal individuals and of individuals with malignant cells. Because up until now, this has only been demonstrated in tissue culture cells. So fortunately, we had a bunch of antibodies that we could show were highly specific, because we could use them on test cells where we can degrade the specific protein of interest. So what you're looking at here as little green blotches are the signals from immunofluorescence for transcriptional coactivators, MED1, BRD4, for a splicing factor, SRSF2, for a heterochromatin factor, and 2 major scaffolds for the nucleolus, FIB1 and MPM1. And in this field, what you're able to do is you're able to take the scaffold proteins, those proteins you think are playing an important role in creating these high valency, these large number of interactions for additional substrate proteins, and show that they can form phase-separated condensates in the living cell, and actually, excuse me, in the test tube. So these are highly purified proteins that will reconstitute through liquid-liquid phase separation droplets, and they give you an assay. And the assay is simply, do various small molecules drug small molecules are typically 500 Daltons or less. Do these small molecules simply diffuse through such condensates, or are they preferentially partitioned into or excluded from these droplets? And what you see here with this dye, fluorescent, and we've seen with Hux and anything smaller than 4 kilodaltons, is that there's a free path to diffusion by these small molecules through the condensates. There's no special concentration of the drug, and there's no real exclusion of the drug from these bodies. By contrast, drugs that are used clinically will concentrate in specific compartments. And in this case, we're looking at mitoxantrone. Mitoxantrone is not a widely used drug. It's still used for prostate cancer. It's a fairly toxic drug. And one of the reasons it's toxic is because it will accumulate in the nucleolus, as you see in the top, where these nucleoli are marked by Fib1 GFP. And the mitoxantrone's naturally fluorescent, and so we'll just color it red, and that's what you're seeing in these cells, these tumor cells. What we see when we use these droplets is that mitoxantrone will concentrate itself in Fib1 and NPM1 droplets. It also concentrates itself in Med1. Now, we wouldn't see that fluorescence in the Med1 condensates in these images because the nucleolus is at least an order of magnitude larger and much more stable than these short-lived mediator-containing transcriptional condensates. But this just says both in the cell and in a molecular representation based on Fib1 and NPM1 of a key scaffold in the nucleoli, you get enrichment. This enrichment looks like it's about 5 to 7-fold. It's understated because the fluorescence is quenched. You have to do other kinds of assays to actually quantify the degree to which that partitioning is occurring. We've done that with cisplatin. Cisplatin is really interesting. The platins are used in one cancer patients worldwide because it's so effective. It platinates DNA, suppresses transcription, affects, it is believed, the ability of tumor cells that are undergoing rapid division to repair their DNA. And what you see here is that it's being concentrated in Med1 droplets in vitro. Now, and the quantification shown over on the right, actually when you use other techniques, mass spec, et cetera, to quantify the amount of partitioning that you get with cisplatin, it is a 600-fold enrichment inside those droplets relative to outside the droplets. Now that's interesting, and we wanted to ask, is it the case that cisplatin binds tightly to Med1? Cisplatin's target DNA is not in these Med1 droplets. So it's being concentrated by some property independent of its target. And what these FRAP experiments tell us is that it's highly mobile, and that can suggest many things, but one of the things it can suggest are weak transient interactions of the type you get with salt bridges or pi-pi or cation-pi interactions. And that led us to consider the possibility that we could infer what we think of as the pharmacokinetic code might be for these Med1 condensates. And we were able to team up with a really talented South Korean chemist, Young-Tae Chang, who built off of a core BODIPI fluorescent molecule various kinds of chemistries with R groups in which he could introduce functional groups from chemistry. And we could use those to screen in vitro these Med1 droplets to discover that in the case of this library and these droplets, what biased incorporation were the presence of aromatic rings. And so that did confirm that interactions, pi-pi type or cation-pi interactions were likely occurring. Whereas the compounds that were dominated by non-aromatic rings were very weakly incorporated into these MED1 condensates. We could take that and we could just ask if it's a pi-pi or pi-cation interaction, perhaps we could remove the aromatic amino acids. And I'm just showing you positive data here. We removed basic amino acids and a variety of amino acids. This is the one that worked. We could remove the aromatic amino acids from the MED1 IDR. And MED1 was still perfectly fine in forming condensates, but it would no longer incorporate those top hits from that chemical library. And moreover, it no longer incorporated cisplatin. So something about the presence of these aromatic amino acids in MED1 was necessary for this preferential partitioning of these aromatic compounds, cisplatin and these aromatic ring-containing compounds into the condensate. So we did a test in vitro. We took advantage of the fact that DNA is conveniently incorporated into mediator droplets and into HP1 alpha droplets, and just asked, would the concentration of cisplatin in mediator droplets lead to an increased level of platination of DNA over what would happen with DNA in the HP1 alpha condensate? And what we found is that's true, that the rate which DNA was platinated if associated with a MED1 condensate was in great excess of what would happen with an HP1 condensate, where that rate is pretty much the rate at which you'd get with DNA in the absence of any condensate. But most importantly, we could ask, what if we now go into cells and simply investigate whether the cisplatin is platinating DNA preferentially at sites where mediator forms condensates? So what I am showing you here now, and I'll just quickly go through this, these are two different cells for MED1, two different cells for HP1 alpha, two different cells for Fib1, where we're looking at the protein condensates here in green in the middle panel, identifying transcriptional condensates, heterochromatin condensates, and nuclear condensates, and where we're at an antibody that recognizes the platinated DNA, not cisplatin, but the platinated DNA, whether or not that's enriched. And indeed, it is in the transcriptional condensates, but it is not in these other condensates that we studied. Moreover, if we do a form of ChIP-seq, where we're using this antibody, let's call it platinum-seq, to identify platinated DNA throughout the cell, what we see is this strong enrichment of the platinated DNA in the superenhancers in these tumor cells. If we allow this reaction, if we allow the cells to be exposed to platinum for a long period of time, then as you might expect, what happens is we lose these MED1 condensates. You can see down here in the bottom left, but we don't lose the heterochromatin condensates in these experiments. So ultimately, that concentration of cisplatin leads to platination of the superenhancer DNA, we believe, including DNA at the MEK oncogene, and that ultimately leads to the destruction of that superenhancer. So this is not a feature specific to cisplatin. This is a feature to all the cancer drugs. This is tamoxifen, fluorescent labeled tamoxifen, or the tool compounds that we've worked with Bradner and Gray that are inhibiting various cofactors for transcription. They are being concentrated in MED1 condensates typically, and occasionally in other condensates, but they're concentrating there by properties that are independent of their target molecule. Even this BRD4, in this particular instance, this BRD4 does not contain the bet bromodomains that JQ1 binds with high affinity to. Similarly, THZ1 is targeting CDK7. There's no CDK7 in these condensates. So what we think is happening then is that these efficacious anti-neoplastic drugs are concentrating perhaps as much as 600-fold in the compartments where oncogenes are active. And this better explains, I think, why we can see these very cooperative kinds of effects where the largest superenhancers driving oncogenes in these various tumor cells are preferentially affected by these various kinds of inhibitors that inhibit transcriptional activity. So how are these new insights going to impact drug discovery and development? I don't know, but it now seems obvious to me that we need to understand how the amino acid side chains are playing roles in promoting the formation or dissolution of condensates, and how the various kinds of post-translational modifications that occur on proteins impact the kinds of interactions and partitioning that we're looking at. I think we may learn then how to target drugs to specific cellular compartments and avoid others, and thereby improve what's called the therapeutic index, the efficacy of the drug over the tendency of that drug to be toxic. And I think what's happening here is a new ability to about how we might take a drug. For example, a chain terminating drug against SARS-CoV-2 transcription replication complexes. And by concentrating it at the appropriate site, allow us to get a very different pharmacodynamic behavior. And it may allow us to target undruggable proteins where we don't have a specific cleft for the drug to act, but rather we can change the condensate behaviors. And we've already seen instances where we can explain drug resistance in cells. This is just meant to remind me to tell you that there are two forms of tamoxifen resistance in breast cancer. One is associated with mutations in ER, where ER no longer binds with normal affinity to tamoxifen. But the other is overexpression of MED1, which makes these condensates larger. It dilutes the tamoxifens available to the cell and makes it less effective. And this is a terrible, this form of tamoxifen resistance gives very poor prognosis. I just gave you this example. If inside this NSP12, that's the RNA-dependent RNA polymerase, inside this condensate where at the moment we know it's a puncta, we can reconstitute the activity of the RNA-dependent RNA polymerase, and that's a condensate in vitro. Can we derivatize chain terminating drugs to concentrate them on target? So I want to end just by thanking some really, really remarkable people. The folks whose names in red up here really led much of the work I talked about today: Ann Boisia, Isaac Klein, Lena Fayen, Alessandra Delagnesian, Susanna Wilson-Hawken. Susanna got her initial training in Tony Koleski's lab. Thank you, Tony. And I just want to give a shout out to Tony Lee, long been a partner of mine in the laboratory in helping us train young people and mentor them into careers in this space. So with that, I'll end and ask for questions.
Thank you so much. You can type your questions in the chat, and I think if you can raise your hand, I should be able to see that too, but I'm going to start with the first question if that's okay. So in terms of the third part of your talk, you talked about where drugs distribute within a cell is really important. I'm wondering, to what extent do pharmaceutical companies, do they look at that now when they're screening for different drugs? Like looking at, do they really partition within the nucleus or anything like that? Do you have a sense for whether they use that in their pipelines.
So they're beginning to, and one of the companies I disclosed on my third slide was DuPont Therapeutics, and that was a company that Tony Hyman and I started in order to actually exploit condensate behaviors and see if there were not new paths to drugs. But there are many other companies that have been set up to do this, and there are many companies that are trying to learn whether or not drugs they've developed are working fortuitously in this way, and more to the point, to learn whether or not drugs that they've been developing, which they think have high affinity for their targets, but once in cells have very low target occupancy, whether this might explain those failures.
There's a question from Antonio Heraldez. I was surprised how many of these molecules were incorporated on Mediator. Is there a specific structure that makes that interaction possible?
Yeah, Antonio, we were surprised too. And I don't know the answer to that question. We're currently screening a much larger library to gain insight into what the partitioning behaviors are of a larger group of molecules. And of course, we'd like to do more of what we did with these mediator mutations to see if we can identify the types of amino acid side chains that are participating. So that's a long-winded way of saying we don't know.
And there's a question from Yu Bao Cheng. Why is Mediator condensate so special? Lots of transcription factor drugs can be incorporated to, oh, this is a similar question, Mediator condensate, but not other condensates. Is there any special feature in the IDR region of the mediator?
Yeah, well, I'll just add to one thing. So mediator is a cofactor for enhancers. So for cell type specific elements. And it is a very large multi-subunit complex that is contributing probably in many ways to transcriptional control. There's a lot of post-translational modifications that occur. One of the striking things about Mediator is it is chock-a-block full of disordered regions. So it may be that it's evolved to interact with a lot of different kinds of side chains in various molecules. And we'd like to see if that's the case. Maybe there isn't a single kind of site we should be looking for, but all kinds of sites that would contribute to electrostatic interactions, to, for example, the pi-pi interactions I mentioned, which is another kind of electrostatic interaction, or to hydrophobic interactions. So there are many are now focused on that might give us insight. And I should just say, at Yale, when I was a graduate student in MB&B, early on, I was not that interested in biophysics. And it was Tom Stites, the chair of my thesis committee, who set me right. And actually, in retrospect, that was one of the most valuable things that happened to me, because now I have a language I can use to discuss with the physicists at MIT and elsewhere what kinds of physical chemical things are going on here and how might we discover it. So thank you.
There's another question about IDRs from Ryan Chow. If most transcription factors have IDRs, how do transcription factors form complexes with some transcription factors, but not other transcription factors? What provides a specificity? Yeah,
great question. So the DNA is still providing specificity, but there's some very interesting work recently that is arguing that really what you're getting is a mixture model where the DNA helps provide the site specificity. A lot of drinking buddy transcription factors get together at these sites that are sitting side by side. You know, if you look at this famous Tom Maniotis interferon beta enhanceosome, that's a 50 base pair long core enhancer that's bound by 7 different transcription factors to really side by side all around that helix. And you get a picture of what's happening at the core of an enhancer that's typically an order of magnitude larger. But the other thing that's happening now is that the IDRs of the transcription factors are helping the factors partition into compartments where you have these nascent enhancers being formed. So now the transcription factors can find the loci where they're going to work, not just by DNA binding, but by partitioning into these sites where lots of mediator molecules live.
There's one more question. I think I'll make this a final one. I'm sorry if I'm pronouncing your name wrong, Kaveh Daneshvar. The size of condensates made of the same protein appear to be very variable. What does the size of these condensates say about their function, especially in terms of transcriptional condensates?
Yeah, it's interesting. Size is important here. The larger the condensate, the lower the energy level of the molecules in it. One way to think about this is that the smaller the condensate, the surface tension is higher on a smaller condensate, and the molecules within it would prefer to be in a condensate of lower energy. So you get something called Oswald ripening that creates that. And one of the interesting things to think about is whether or not there are what are called depot effects for most of the functions we're talking about. For example, I now think of nuclear speckles as huge depots for splicing apparatus. And when you get co-transcriptional splicing, there's the formation of a small condensate at the site of transcription that borrows some of that apparatus transiently to get initial splicing done, but then releases it back to the speckle, that much larger condensate, when that reaction is no longer occurring.
Great. Is there any questions that I've missed? No, I think that's all we have in the chat. So thank you, Dr. Young, for the seminar today. And thank everyone else for joining in today.
Thank you so much.